For this project, I collaborated with group members Yara Batista, Olivia Legel, and Jack Paras. The title of our project is The Effect of Cranberry Juice and Sigma T Extract on Apoptotic Induction of HeLa Cells. HeLa cells have been around for nearly 70 years now, and they have been used in studying numerous things like human diseases, and these cells have also even been the first isolated cells in space, and have even gave way for many new vaccines and treatments, and also could be responsible for one day eliminating cervical cancer as well. In this study, we look at the anti-proliferative effects of cranberry juice on HeLa cells, looking closely at cell morphology and the blebbing of the nucleus. Along with cranberry juice, HeLa cells were treated with a positive control, which was DMSO or dimethyl sulfoxide. They were treated with a negative control, which was colchicine. We also treated cells with sigma T as well, and it has been known in previous studies to induce apoptosis. The results of our experiment showed that HeLa cells treated with cranberry juice decreased cellular proliferation, but not in a significant manner in comparison to DMSO. Cancer is extremely prevalent in society today, and we are always looking for better treatments with less of the negative effects. These negative effects typically are a result of treatments killing healthy cells as well as the cancerous ones. Previous studies show that many polyphenols derived from natural compounds have the ability to disrupt the microtubular network within cells, causing cell arrest and then apoptosis. Our big question is, what other natural compounds besides the known are able to inhibit cell growth by targeting the microtubular network? Previous research has shown that many T polyphenols does just this and leads to an arrest in cell cycle, thus inducing apoptosis. One of the polyphenols in black tea, theaflavins, does just this, and the Chakrabarty study in 2011 supports this idea as well. We will be exploring the possibilities of other natural remedies other than tea compounds inducing apoptosis, like cranberry juice. Previous studies have shown that cranberry derivatives cause a decrease in cell density, but not many studies have explored the effect on cell morphology. Our specific question in this experiment is, do cranberries and cranberry derivatives induce apoptosis in the same way previously researched natural compounds do? We predicted that cranberry juice will induce apoptosis in HeLa cells, and that HeLa cells treated with cranberry juice will appear more fragmented when viewed with fluorescence microscopy. Now on to our materials and methods section. Taking a look at the material and methods, there were 6 big steps taken in order to view our HeLa cells. In this first step, we treated our cells with our 4 intermediates, DMSO, colchicine, sigma T, and cranberry juice. Next, we fix our cells with paraformaldehyde in order to freeze the cells in their current state. Then, we permeabilize the cells with Triton X100 detergent to make the cell membrane more porous in order to facilitate adding our antibodies. Next, we then added our blocking solution, which was 2% BSA, then added our primary antibody solution, or antitubulin TRITC phalloidin. Then, in order to view our cells, they were stained with DAPI. DAPI allows us to get to our last step since it is required for cells to be viewed under the fluorescence microscope. Now into our results. Figure 1 shows HeLa cells captured with fluorescence microscopy after treatment from our 4 intermediates. As expected, colchicine killed most cells and DMSO allowed cells to thrive. Like other experiments have also shown, sigma T was effective at killing apoptotic HeLa cells. As for cranberry, although there are some cells that underwent apoptosis, the number was comparably less than both colchicine and sigma T. Now, looking at figure 2, in this figure we used collected data from cell counting and displayed it in a bar graph. This bar graph shows the average number of apoptotic and normal cells in each of our samples. As we saw in figure 1, everything went as expected except cranberry apoptotic cells are much less than colchicine and sigma T. We then conducted 2 two-tailed t-tests and found that there was a significant difference between the apoptotic values between DMSO and sigma T, and there was not a significant difference between DMSO and cranberry juice. To summarize our results, looking at figure 1, there was a very small increase in apoptotic cells for the cranberry treatment. Colchicine and DMSO sample results stayed true to their controls, and sigma T had an intermediate amount of apoptotic cells in comparison to both colchicine and cranberry. The important findings in figure 2 came from our t-test, which once again showed that there was a significant difference between the apoptotic values between DMSO and sigma T, and there was not a significant difference between DMSO and cranberry juice. Now on to our discussion. Our hypothesis that HeLa cells treated with cranberry juice would induce apoptosis was not supported, given the results in t-tests conducted. We noticed a change in cell morphology after the sigma T extract treatment, along with the presence of bleb nuclei. This was a prevalent theme as well in Chakrabarty's experiment, which found that apoptosis induction is responsible for alterations in cell morphology after HeLa cells were treated with theaflavins. To reinstill why our hypothesis was not supported, the t-test conducted between DMSO and cranberry comparing apoptotic values was greater than 0.05, meaning there was no significant difference in the number of bleb nuclei. This suggests that the apoptotic cells in the cranberry treatment could have been due to chance. A study conducted by Wei and others showed that a cervical cancer cell line treated with cranberry extracts were inhibited, but no apoptosis occurred. This suggests cranberry might not be effective at killing cells, but inhibiting growth. But this same study also found that breast, lung, and ovarian cancer cell lines with high concentrations of cranberry extract cause apoptosis in these cells. This could mean that cranberry extracts work differently in different cancer cell lines, or there might be some experimental error giving different results. While our experiment was unsuccessful in showing that cranberry can significantly induce apoptosis, we think that extracting the derivatives out of cranberry juice and applying them in high concentrations might convey different results. The study by Wang showed just this, as the extracted polyphenols he used did lead to induction of apoptosis. I wanted to give a special thank you to Dr. Llewellyn for facilitating our learning and understanding of HeLa cells in the lab, and for making this transition to online much easier while still allowing us to learn at our full potential. Thank you.